Hello everyone, welcome to our presentation of Onkopus. My name is Nadine Kurz. I'm a member of the Department of Medical Bioinformatics at the University Medical Center Göttingen, and today I'm going to present our decision support framework for evidence-based interpretation of actionable biomarkers in precision oncology. The goal of precision oncology is to identify new treatments that directly target cancer variants. For this purpose, we aggregate information from public databases where the effect of each treatment on a cancer variant has to be proven by at least one study to ensure evidence-based information. The results of the variant interpretation are then discussed in a molecular tumor ward, which is an interdisciplinary committee which discusses each cancer patient on an individual basis and makes a retreatment recommendation. The MTB workflow may be summarized in 5 steps. After a tissue sample of a cancer patient is taken, rare genetic variants are identified. The most extensive task is then the variant interpretation procedure. The results are discussed in the MTB conference and a treatment recommendation is made. As there is a high need for computational support for the variant interpretation, our goal is to support the interpretation by providing annotation and focusing on the prediction of variant pathogenicity and the prediction of clinical significance of available treatments. The main components of Onkopus are the Onkopus server and the Onkopus modules. The server may be accessed by a web application called Onkopus Web and a command line interface, which is a Python package. You may query in Onkopus Web for a biomarker directly or upload a full variant file like a VCF, a MAF or CSV file. The main feature of Onkopus is its modularity. Each Onkopus module is a container which has its own RESTful API. The Onkopus server then accesses the modules. Our goal is to provide modules for each feature of a variant, including biochemical features, population frequency, drug-gene interactions, pathogenicity scores, functional effect prediction, and clinical evidence data. We then aggregate and normalize this data and provide a final score for the pathogenicity and the clinical significance data. The output of Onkopus is an annotated biomarker file in CSV or VCF format, possible graphs and statistics, a human-readable report in PDF, and a metadata file. As for our pathogenicity prediction, we calculate multiple pathogenicity scores and categorically group them according to their most important aspect, including conservation, protein structure, or machine learning. As for the clinical significance data, we search for different match types. We search for exact matches, like BRAF V600E, other mutations at the same position, or other mutations in the same exon. Finally, we are searching for clinical significance data for any mutation in the whole gene. The clinical significance data is then visualized in a sunburst plot, which gives us the possibility to visualize multidimensional data in an interactive and zoomable way. Here we visualized the drug class, the drug, the evidence level, and the PubMed ID. Let's take a look at the web application. After accessing the start page, you may click on Start. Then you can upload a file directly or search for one or multiple biomarkers. You may enter a gene name and a protein change, a genomic location, insertions and deletions, or a gene fusion, where the Onkopus server automatically recognizes the request types. If you click on Search, you get possible plots, including the biomarker positions and clinical significance data on all variants, and a result table, which includes the mutation type, gene name and protein change, ClinVar estimations, the most important pathogenicity scores, and the number of possible treatments found. If you click on Details, you may get a detailed view of that variant, including a summary of gene information, population frequency, ClinVar estimations, variant information, and genomic data. If you click on pathogenicity, you get the already shown categorically grouped pathogenicity plot and some of the calculated pathogenicity scores. If you click on treatments, you receive 2 clinical significance plot and the full result table. If you click on the first plot, you may browse possible treatments according to the drug class, the drug evidence level, and PubMed ID. You may click on each cell and browse the full dataset according to these features. If you close it and open the second plot, you get the same results, now plotted according to cancer entity, evidence level, drug, and PubMed ID, where the color indicates the response type, like sensitivity or resistance. In the result table, you get a full list of possible treatments and the match type, including the PubMed ID and evidence statements. Additionally, we want to thank our supporters, including the Comprehensive Cancer Center Niedersachsen, the Volkswagen Stiftung, and the International Max Planck Research School for Genome Science. That's it for now, and thank you for watching.